Welcome to the very first episode of a new journal club that I'm starting, entitled The Classics of Immunology. And this has had a long gestation and birthing period. It started really 15 years ago when one of the postdocs in the lab wanted me to buy him a new textbook of immunology. It was really an expensive textbook. And I said to myself, I didn't tell him this, but I said to myself, that's a waste of money. But what I said to him was that, the only way, and he was a biochemistry major in the school, and he got into immunology secondarily, and so he didn't have a clue what we were all talking about. You speak in all this jargon and acronyms and all kinds of things like alphabet soup. And so I said to him, the only way you're really going to learn immunology is that you have to go back and start at the beginning and read all the classic papers. And so that gave me the idea that what we should do is instead, we were having a journal club like every lab does, that would once a week, one of the members of the lab would give a presentation of a recent paper that came out that was of interest to the kinds of things that we were doing in the lab. And I thought to myself, well, what we should really do is we should change our journal club so that we really focus down on the classics of immunology. And so that's what we did. And over the course of several years, we read all of the papers that I could think of that were the classics of immunology. And as time went on, what I did with those papers was that I put them on my website. So if you go to my website now, kendallasmith.com, and then click on the tab for writing, what you'll find is first the books that I've written so far, and then selected papers of our papers from my lab over the years. And then starting after that are the classics of immunology that are listed there. And they go on and on and on. I didn't count them, so I don't know how many I've got there, but must be at least 200 or so. So that's what we're going to do. We're going to start at the very beginning of immunology, which was in 1798 with Edward Jenner. And I've talked about this paper before, and so I don't want to be redundant and go through all the things that I've already talked about. But there's some very salient points that Edward Jenner established in the very first paper of immunology. Essentially, there wasn't anything like this until he wrote this paper, and it's really not a paper, but a monograph. And I have it right here, and I'll show it to you. You can read that. And so there you go. It's an inquiry into the causes and effects of variole vaccinae, a disease discovered in some of the western counties of England, particularly Gloucestershire, and known by the name of cowpox. And so this was the beginning of vaccination. And because of the fact that we are now embroiled in a worldwide pandemic of a viral disease, coronavirus, there's a lot of points that Jenner made in his paper that are really important for us where we are today with coronavirus. And so I thought I'd just talk about those a little bit. The monograph is, so it's not just a short paper like we have today in science. It's 75 pages in this book. So this is a book, it's like that. And he also had, and he self-published this. He went to a publisher in London and got them to put it out. And he also had an artist do lithographs. And here's one of a vaccination of using the cowpox virus in the arm of a vaccinee. So it's very, very interesting. And I recommend if you follow along with this journal club, you should read all the papers, and you can get them off of my website. They're all there. So you don't even have to look for them very much. And I wish when I had started out in immunology, because I first went through medicine, and I didn't really know a whole lot about immunology, because when I started out, it was just really at the beginnings of the modern science of immunology. And had I had this, I could have hit the ground running. But whereas it took me like 20 years to figure all this business out that we're going to do in this journal club. what this monograph is a compilation of case studies. Now, case study in medicine is a description of a person's disease, what they present with to the doctor, and then a detailed analysis of all the signs and symptoms and laboratory values and all these different kinds of things that allows the doctor to make a diagnosis. And you don't really write up case reports of regular old mundane things. It's usually a very unusual kind of a situation. And so what Jenner did, and he had 23 cases of either smallpox inoculation, where you take a little bit of the actual disease, smallpox, from a pustule and inject it under the skin of a susceptible individual, and you hope that he becomes immunized and it doesn't get a disseminated infection and die. This would be a bad thing. And smallpox, for the last 300 years, has accounted for about 500 million people dying worldwide. It's huge. It makes this coronavirus look like this is just a little flea in the ointment here. And the thing about smallpox is that it wasn't a pandemic that they were dealing with back in the 18th and 19th centuries and 20th centuries, but it was an endemic problem, so that it was basically there all the time. And, smoldering levels, wasn't much, every now and then there'd be a case and so forth. But then also every now and then there would, for whatever reason, it would change from endemic to epidemic, which meant that it would go through a village and all the susceptible people would get smallpox. And that usually sorted out between the young people who, because they hadn't been exposed to smallpox before, versus the adults and the older people who had lived through epidemics of smallpox and either had gotten the disease or hadn't gotten the disease, but had been exposed to the disease. And it was well known that if you got smallpox and didn't die, it came with a 30% mortality. And just to compare that to what we're dealing with coronavirus, only 2% of the infected people or the reported cases have died. So smallpox, it was 30%. And also, if the virus got into the cornea of your eyes, it basically had a major inflammation that left you blind. So it accounted for most of the world's blindness back in those days. So it wasn't a good thing to have. What happened was, in the history of this, and Jenner goes into it a little bit, but what happened was that in Asia, in China and in India, people had started taking fluid from the pustules of one patient with smallpox and then injecting it under the skin of a susceptible individual. And what happened was that most of the time, the people would become noticeably ill. Usually took a week or more, but they would then ultimately recover and go on, and then they were essentially immune from this whole thing. And what Jenner did was he improved upon that, because he didn't use smallpox to inoculate into the individual. He used a virus that was taken from the udders of cows that was named cowpox virus. Of course, back in 1798, they didn't know what a virus was, and it wasn't until after World War II we finally had a picture of a virus because of the electron microscope. So if you look in the dictionary, virus just means, in Latin, it means poison. Anyway, what would happen was that the milkmaids in the dairy country west of London, Gloucestershire, the county, was a dairy farming community, and everybody knew in the dairy farming community that if a milkmaid it's sort of like one of these gender discrimination sorts of things. Only the women milked the cows and the men took care of the horses. And this is important from the standpoint of the history, in that what would happen is there'd be horses and cows in a given farm. And horses, particularly in the springtime, would get an affliction on their hooves, so that they would get all inflamed and weepy with pus and so forth, and it was called grease. And the men took care of the horses, whereas the women took care of the cows. But the men every now and then would be drafted into milking the cows too, when they had when they had a rush or whatever. And if they were then transmitted the virus from the horses to the cows, and then from the cows to the milkmaid's hands. And I have a picture of one of the milkmaid's hands from Jenner's monograph here. And so, you get these pustules, so they look like smallpox, but the difference between cowpox infection and smallpox infection was that smallpox would become disseminated all over the body, and you'd have these pox kinds of lesions. And if they became confluent, you really got sick, and then you would ultimately die with a systemic inflammation, essentially, which is very similar to what's going on with coronavirus today, because the symptoms of coronavirus are not due to the virus. They're due to the immune response to the virus. So high fevers and shaking chills, and ultimately tremendous inflammation in the lungs, so that the lungs fill up with inflammatory fluid. That's why you die with coronavirus, and it's all due to the immune response. In all of these different case reports that Jenner put into this monograph, and that's why it's very interesting if you want to become a scientist to read this, is because he was very, very careful about how he described each of the cases and his interpretation as to what that taught him about the whole process. And so the first point that he made is that it probably went from horses to cows to people. And each jump in a species seemed to be important for the generation of this virus or viral fluid that he used then to try to inoculate into susceptible individuals. The second point that he made is that one of the things that would happen is that you didn't even have to have contact with the horse's hooves or the cow's udders and the pustules there. You could get it from fomites. Fomite is a word that doctors use to describe, usually innate objects that people have touched and left the virus behind. And then you think about a doorknob was the easiest thing to think of. In this instance, it was the milk pail, and all these different people, the guys and the girls picking up the milk pail, could transmit the virus, the cowpox virus. In the process so Jenner was just like all the rest of the physicians. He was a physician, a country doctor essentially in west of London, and like everybody else at the time, people had started inoculating smallpox into people, and doctors had inoculation clinics so that they would bring people in. They'd inject them with smallpox, and then when they got sick, they would care for them afterwards. And so it was sort of a tremendous money-generating operation. You can imagine these guys. So if you weren't sick by yourself, come on in my clinic. I'll make you sick, and then I'll take care of you. And different doctors boasted about their mortality rate, and if it was down by like 1% instead of 30%, this was a good thing. So you were taking your chances. But if you waited for an epidemic, and this is where the parents would come into play, they're scared to death that their kids were going to get this thing and die. Was a strong probability. So it wasn't until case number 16 in Jenner's monograph where he, and that was the picture of the hand that I showed you. From the person's name was Sarah Nelms, and she was a milkmaid, and she got cowpox on her pustules on her hands in May of 1796. Jenner took the fluid from one of those pustules, and he inoculated case number 17, which was an 8-year-old boy, on May 14th in 1796. And about 7 days later, so the boy was fine. About 7 days later, he got a swollen, tender lymph node in his axilla, his armpit. And he had a slight fever, and he had generalized aches and pains and didn't feel so well for about 24, 48 hours. But then after that, and he got a pustule on his arm, like the one I showed you on that picture. And then everything subsided over time, and he was fine. So then Jenner, and this is the critical part, then he did the next thing, and that was, but he wanted to know whether or not this child was really immune to smallpox. So, as I've said before, is that one way to find out is to challenge them with smallpox, which is what he did. And basically, nothing happened. The child was fine. So then Jenner, and this is another part of the whole story that I find quite amazing. So then Jenner started wondering whether or not the stuff that he used to challenge with the smallpox was really, a viable preparation and so forth. And so he did it a second time several months later. And again, nothing happened. And at the same time, he took the same preparation and inoculated other people, and they got what they called a take, which was what the skin looked like on that boy's arm that I showed you. Then he went on did exactly the same thing in several other patients, and in one situation, and he generated case 19 and case 20 and 21 and up to case 23. He took the material from one individual, and he then inoculated several people with that same preparation, and he was able to show that you were able to pass. Well, no, he would give to one individual, and then he'd take stuff from that person and put it into another one, and he could pass it on to several, iterations of this whole thing, which is important because that's what happened. That's how they ultimately developed the vaccination that Jenner then spread worldwide. He collected all kinds of pustule fluid, and he'd sent that off to Europe, and then ultimately the States, and then ultimately into the Far East. And he became really a rock star worldwide because of this. In his discussion of all of this, he made a couple of other points that were very important in terms of what happened down the line and why this is really a classic paper. He speculated that the disease, that the generation of the attenuated vaccine virus, which ultimately took the name of Vaccinia, and that goes back to the French, because the French word in Latin is vache for cow. But he thought that it came from the horse and the grease on the horse's hooves that had to go into cows and then before it would go into people. And that's interesting because that happens in other situations, with other viral infections as time has gone on, and we've had more experience with this kind of thing. And what probably happens is that the virus mutates and becomes adapted so that it can become infectious for different species. And of course, that's what the story is with the coronavirus. Back to the coronavirus, since everybody here has been educated about coronavirus on television and so forth in the nightly news, they think it came from the bat and went into a human, and that's very feasible because the bat is a mammal, and mammals and humans are mammals. And then he goes in his monograph, and he described very carefully how to distinguish a pustule from smallpox and also from Vaccinia as compared to other kinds of pustules that would develop on the skin of individuals. And of course, now we know this is 100 years before other bacteria were found to cause all kinds of infectious problems, one of which being you'd get carbuncles and boils on your skin that would fill up with fluid. Looked very much like these pustules in smallpox and cowpox. And so you didn't want to transfer that stuff into say an 8-year-old boy. And one of the reports in the literature that was sort of really terrible is that they took some material from somebody who had secondary, tertiary syphilis and inoculated a little boy with that. And of course, then the poor little kid got syphilis. Not such a good thing to have happen. And so he was trying to educate the other doctors as to how to distinguish between the good stuff versus the bad stuff. He also had worked out a very meticulous method of injection, inoculation into the skin, and he taught that in this 75-page monograph. And the other thing he showed is that the cowpox virus really was not transmissible between humans. It wasn't like smallpox could go from one human to the next human to the next human. Cowpox did not, and the reason that is that cowpox was a self-limited infection. Smallpox, which is very much like coronavirus, is a respiratory transmission. So it comes, you breathe it in, it goes into your lungs, and when it goes into the lungs, it's distributed throughout the lungs, and then the heart pumps it out to all the tissues in your body, and that's why with smallpox you get disseminated infections or disseminated pustules all over the body. So all of those points that Jenner makes that really the classic paper of immunology, and everybody should read that paper. But I think the other thing that I'd like to accomplish in this journal club is to try to relate, what was really reported in the original paper and how that relates to what we know about things today. And in this particular instance, I've studied this quite carefully, and I've written several papers about Jenner and the smallpox thing. And one of the papers that I was asked to write a paper by the journal, the Indian Journal of Medical Research, because they were having a 100-year anniversary, and they asked me to write a paper about the eradication of smallpox and how that came about and so forth and so on. So I wrote that paper, and if you're interested in it, it came out in the Indian Journal of Medical Research in May of 2013 in volume 137 and page 895. So you can look that up in your library. And it's entitled Smallpox, Can We Still Learn from the Journey to Eradication? The importance of this whole business, as I alluded to just now, was that it was transmitted through a respiratory route. And just like coronavirus, it led to a pretty long so-called incubation period of 10 days. And it turns out that now that we've done a lot of more science, the smallpox virus is a big virus. It's a double-stranded DNA virus, and it has more than 200 genes in it. And just to compare that to us, we have about 30,000 genes. So it has 200 genes, but about half of those genes they encode for molecules that inhibit the innate immune response, the first responders, which mostly have to do with interferon. And interferon interferes with viral replication. And so once a cell becomes infected with a virus, it triggers the production of interferon, and then the interferon is released by the cell and binds to receptors on other cells and imparts an inability on the part of the cell to become infected and to reproduce virus. So interferon is really important. And what happens in smallpox also happens in coronavirus, which is that the virus inhibits the innate immune response. And that's why you don't have any symptoms. Because as I said, the symptoms are not due to the virus. The symptoms are due to the immune response to the virus. And it's exactly the same thing with smallpox for the same reason. Because the virus is replicating all over the body after it goes in through the lungs. And it's sort of like, they're home free. There's no impediment. There's no natural innate immunity to these viruses. So, and then in the end, what happens exactly the same way is that then after 10 days, 10 to 14 days, the adaptive immune response that's mediated by T cells and B cells gets into the action. They make a lot of cytokines that are inflammatory cytokines, and ultimately you end up with disseminated systemic hypersensitivity kinds of phenomenon, and that's why you get sick and die in both of these infections, essentially. Then the question was, smallpox is interesting for another reason, and that is that it's the only infectious disease that has been eradicated. The virus has been eradicated from the face of the earth. And the immunologists and all the medical people, they like to point to that and say, this is what we did for you. It certainly is good. And the question is, what's so special about smallpox that allowed it to be eradicated? And there's several parameters about smallpox virus that made for a situation where we as human beings were able to do that. The first is that it was easily diagnosable. And 100% of the people, it wasn't like coronavirus where you can have these asymptomatic people spewing out virus and infecting other people. It wasn't until you got those pustules, and then they would break, and then they would release virus, and then just in the proximity of everything, other people would become exposed to them. They would breathe it in, and it was transmitted respiratorily. But with the rash that you got afterwards, within about a couple of 3 days later, after your fever and so forth, it was easy then, or as it was possible, to educate healthcare workers, as we call them today, to scan the community and report cases to the authorities of people that had smallpox. And then they could send people to interview those people. First of all, quarantine those people, and then interview them to find out who their contacts had been during that time. And for the most part, people were, in contrast today, most people were pretty sedentary in their lifestyle. Most people would grow up and die in the same little village that they came from. And so that's basically the contact tracing that we've been talking about. So that could take place. And then those contacts, teams of people would go out and immunize them with the vaccine. And as they did that, they set up a thing in the 60s where they went worldwide, country by country by country, the public health people and so forth, and eradicating smallpox from the country, essentially. And it took them about a decade, but they were able to do it and satisfy themselves that there was no more smallpox anymore. The other thing about smallpox is that there's no animal reservoir, like bats, for example, that would continue to seed the human population. So it was only a human disease that they had to deal with. And the final thing is that they had developed since between 1800 and 1970, they had developed a very inexpensive, stable vaccine preparation that was could be stored at room temperature. A lot's been said about the new virus or the new vaccine that's come out from Pfizer BioNTech, and it has to be held at minus 80 degrees centigrade. That's sort of a problem if you don't have one of those kinds of freezers. But the Dryvax is what they called it eventually. And the way they initially got it was they would inoculate the skin of calves until there was a disseminated infection on the skin. And then they would sort of scrape off the material and then purify it and basically figure out the concentration of the virus, and so that they had a stable vaccine that they could send around worldwide. And so all of those characteristics of smallpox made it possible for them, for us, to get rid of that whole disease. And the other thing about it is that because it was endemic and then epidemic, a lot of people were already immune because they'd been survived other epidemics and so forth, or endemic situations where they got an infection. But then once they started vaccinating, they could test how many people were vaccinated. And the Vaccinia would leave a scar on your arm or wherever you got it. I think they gave it to little girls in their bottoms, and they gave it to boys in their arms, and it would leave a scar. And you had to show that in order to go into the first grade. Otherwise, they wouldn't let you in school. So even if there were religious anti-vaxxer dissenters in the whole situation. But they talked about, and this is very important, they talked about the percentage of people who became immune within the population that was necessary to give the so-called herd immunity. So that if you got and so it was upwards of 80% in smallpox, and in measles it's 90%. And Tony Fauci, who's become godlike in our business now, was on TV just yesterday saying, early on he came out and he said, well, we're going to have to get to 60 to 70% people within the population to have herd immunity, so that the herd protects those susceptible people from becoming infected. But now he's changed that just yesterday, and he's saying it's probably going to be close to 90% we need for herd immunity, which I think what that's going to leave us essentially is we're not going to achieve herd immunity, even with the vaccine, because we have the anti-vaxxers in this world who don't want to become vaccinated, are afraid to become vaccinated, don't trust the medical profession and the scientists and so forth. So I'm going to be surprised if we get over 60% vaccinated in our country. I don't think it's going to be, we're going to be lucky if we get higher than 50%. And what that's going to mean to us is that we're going to be left with an endemic coronavirus. It's just going to be here all the time. There's going to be a certain fraction of people who will become infected, just like with the common cold. And most of those people will recover, but 2% of them, according to the numbers that we have now because of the pandemic, 2% of them will die. And that's just going to be life as we've lived it. And so if you choose not to become vaccinated, you have a very high chance of becoming infected because it's endemic, it's every place. And of those people who are infected, you have a 2% chance of dying. So I forgot to say, there's one other thing. The reason they called the vaccine dry vax is because they could freeze dry it. They could lyophilize it and then send it out and they have the little vials. They freeze dry the vials. So and they ship it all over the world. And so then all they had to do is add fluid to it and draw it up in a syringe and inoculate or inject the next person. So that's the story with Edward Jenner and the cowpox vaccine. And I hope you guys get a lot out of this journal club. I'm going to have fun reviewing all these papers myself. Thank you. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.